was really intended to bring the excitement and the potential of immunology to the very broad audience. I just want to tell one story and a couple of personal comments related to Bill. In 2003, Bill was asked to become the editor of Immunity. He asked Ron Germain, Larry Samuelson, BJ Folks, and me to join him, but as has been indicated earlier by his collegiality and generosity, he didn't want to be the main editor. He just wanted to be a co-editor, and we were all formally equal editors, although I think 4 of us viewed him always as the more senior editor. It was remarkable meeting with Bill every week, and one of the things that one could observe is that he always looked for what was good in papers. Was it exciting? Was it new? And then he worried secondarily about whether the data were compelling enough to justify what was being communicated. I never actually worked in the LI, was never really per se with Bill, but he treated me incredibly well, was an advocate for my career throughout. I cherished the many conversations that we had, and he always seemed as if he was available. If one called him with a question or whatever, the call would come back either the same day or maybe the next morning. It just seemed that he always had time to talk to people. And so anyway, I miss him. As Bob Cedar said, he really was a mensch. Marilyn, if you could come up and say a few words, and then I'll introduce Lori.
Afternoon is thank you. And first and foremost, I must thank Dr. Ron Germain for coming up with the extraordinary idea of creating an all-day seminar dedicated to Bill's memory. This is just the way Bill would like to be remembered. And then Pam Schwartzberg took over, and Ron had created a list of eminent scientists, very busy people, people who are in demand, and it was Pam who had to call them and get them all to agree to come here on a specific day that everybody would agree to, and on a day when the auditorium was available. So this was a major challenge, and we owe a great deal to Pam. Other people who made it work, I must mention Jeff Zhu, who was stuck on a slow boat from China, and Josh Milner, Howard Young, Huyen Park, Kathy Esterday, Jane Hugh Lee, and Effie Reyes, who ran the LI lab, the LI office for more than 29 years. And I want to thank this morning's speakers who got the program off to a great start. Thank you so much, Tony Fauci, Harold Varmus. Mark couldn't make it, and I miss him not being here today, and Bob Cedar. And to the trainees, thank you all. You honored Bill years ago by coming to the lab as postdocs and fellows, and so many of you return today to honor his memory. And special thank you to special lecturer Mark Davis, who was also a trainee, you already know that, and to Dr. Glory Glimcher, who is going to give the keynote in a few minutes. She too was a trainee here. And thank you to the Cytokine Interest Group for the William E. Paul Best Paper in Cytokine Research Award. That gives me a great thrill, and that is a great honor to remember Bill's name in this way. And now for the LI. How can I ever thank you enough? Bill inherited a small, weak laboratory, and it was you scientists who came to the LI many years ago over many years and worked your magnificent science and turned the LI into one of the world-renowned laboratories. You fulfilled Bill's dream. You made his dream come true, and I thank you enormously. I'm not sure that I have the words to express what I feel in my heart. And I look out at this gathering, and I want to thank every one of you sitting here who are honoring Bill's memory. Bill loved the NIH. He loved science. He loved immunology. He thought the NIH was a fabulous place to do science, and he thought it was an excellent example of the federal government at work. Thank you all.
It is a real pleasure to introduce Lori Glimcher, and I think for this audience, Lori really doesn't need an introduction, but Lori, you don't get off quite that easily. So, Lori went to Radcliffe and then Harvard Medical School. She was a medical resident at the Mass General, worked for a year with Harvey Kanter, and then came to the LI, where she had a myriad of first author papers, working together not only with Bill, but also with Ira Green, Ron Schwartz, and Ethan Chivak. Afterwards, she returned to Boston as a rheumatology fellow at the Mass General, and then joined the faculty at Harvard, where she rose through the ranks to become the Irene Heinz-Given Professor at the Harvard School of Public Health, as well as a professor at Harvard Medical School. In 2012, she left Harvard to become the Dean of Weill Cornell Medical College, where she was phenomenal with new recruitment, helping to make a strong place even much stronger than it had been. Earlier this year, she accepted the position to become president and CEO of the Dana-Farber Cancer Institute, where she is taking on new challenges and returning to Boston, of course. Her academic career has been stellar, with cloning of Tibet, discovery of CMAF, discovery of XBP1 in the context of immunology, but as she'll highlight today, XBP1 has really allowed her to transition via the unfolded protein response and stress to the topic of her talk today related to cancer. Lori has a CV that is overwhelming. Let me just say that in addition to all the major academies, she's been on the scientific advisory board of the National Cancer Institute for almost 20 years, on the corporate board of directors of Bristol Myers Squibb, and she also serves on the board of directors of the Parker Institute for Cancer Immunotherapy and multiple others. So without further ado and stealing any more of her time, let me welcome Lori to give the first annual William E. Paul WALS lecture on a Monday, but it will be on Wednesdays. Her title is Stressed Out, a Novel Approach to Cancer Immunotherapy.
It is an enormous pleasure and honor for me to be here with you today. And dear Marilyn, dear Matthew and Jonathan, and all of the Paul family, thank you so much for sharing Bill with us. I miss him, and I miss him even more as time goes by. So I'm going to talk today about one particular signaling pathway that we've been interested in for many, many years, whose exploration actually has led us into many different fields, and most recently, as Warren said, into the study of cancer. But before I do that, I do want to say a few words about the man that we are honoring here today. Something that I think nobody has mentioned yet is that Bill was one of those rare individuals who always supported women. He had many female postdocs. We did call ourselves Bill's lymphets, which perhaps nowadays wouldn't go over so well. But Maureen, I think you remember that. And I think many young women nowadays would find it hard to believe how little understanding professional women could receive in those days when they tried to combine career and family. And in that regard, and in many others, of course, Bill was revolutionary. He made great contributions, as you've heard from others, in our understanding of how the immune system works, but he also made great contributions to the scientific and personal developments of the many, many people, including me, who had the great good fortune to work with him. He had an enormous impact on my life, and I will miss him every day. I was looking through my notes, and I found a letter that I sent to Bill when he received the AAI Mentoring Award. And the letter said, "Dear Bill, I'm so disappointed that I cannot be there in person to celebrate with you. You were and are a mentor extraordinaire. I must confess that I was surprised to learn that you won the award because I assumed you must have received it a long time ago. But this gives me the opportunity to say how much you mean to me and how grateful I am for all that you've done. My independent career in immunology was launched by you, and you taught me a great deal about both science and mentoring. The breadth and depth of your knowledge of immunology impresses me to this day, and your service to the immunology community has also been profound. I also threw up in your office in 1981 when I was in the throes of morning sickness, and you didn't blink an eye. Good man." And similarly, today I am so disappointed that Bill can't be here with us in person to celebrate his amazing career and his remarkable life as a human being. But in honor of Bill's mentorship, I decided to organize my talk today around themes that Bill was passionate about and areas that he made major contributions in, while at the same time focusing on only one interest in my own laboratory that's occupied us for the last 2.5 decades. And that is, let's see if I can remember how to use this. Thank you. So, Bill's made many contributions, and I'm going to tell you, oh, there it is, thank you. That's good, thank you. I'm going to tell you very brief stories about these 4 areas in which Bill made major contributions and in which those contributions influenced me. And the first was an MHC class II gene regulation. Bill had discovered that interleukin-4, which of course he was the one to discover, control the regulation of MHC class II proteins in B cells and in other cells as well. And that got me interested from moving from the structure and function of class II MHC molecules, which I had done as a postdoc in Bill's lab, into the regulation of MHC gene expression. And along those lines, a graduate student in my laboratory decided to clone the transcription factor that might regulate the MHC class II DR alpha gene. And she did that using a Southwestern technology that was very au courant at the moment. And the gene that she cloned, we named X-box binding protein 1. It's not a very exciting name, and we can't change it now, unfortunately. And that was because it bound to a promoter region in the HLA-DR alpha gene that was called the X-box. So we set out to figure out what this protein actually did in vivo. Now, in vitro, it clearly controlled the expression of MHC class II genes. But here's another lesson that I learned from Bill. In vivo veritas, in vitro maybe. And it turned out that when we deleted X-box binding protein 1 in mice, first of all, they died from embryonic lethality because of failure to form the liver. But when we made lymphoid chimeras, we found out that what XBP1 did in vivo had nothing to do with the regulation of MHC class II genes. Instead, it controlled the differentiation of mature B cells to plasma cells. And Bill contributed a large body of work on the development and function of B cells and B cell differentiation. And so I have to thank him for inspiring this interest in me. Now note that the resting B cell, which was completely normal in mice that lacked X-box binding protein 1, have small nuclei. But once they become an active B cell, a plasma cell, of course, they expand their ER because they're secreting lots and lots of proteins. Well, if you don't have XBP1, you don't make any plasma cells, as you can see here, and you have no serum immunoglobulin of any isotype. And just to make a nice circle in the story, XBP1 is influenced by interleukin-4. So again, you see memories of Bill in the work that we did. Now, how did we get into the unfolded protein response or endoplasmic reticulum stress response from that beginning? Well, I actually knew nothing about this response until 3 labs independently published that the long sought after mammalian homologue of the yeast factor called HAC1P was XBP1. And now it made a whole lot of sense why these animals did not have plasma cells, because I can't think of another cell in the body that secretes more proteins and has to handle protein loading more than the plasma cell. And the ER stress pathway, which was really discovered by Peter Walter, working in yeast, is a pathway, it's a signal transduction pathway whose goal is to protect cells from the stress of unfolded or misfolded proteins in the ER. It turns out that there are other stimuli that evoke the ER stress response, and we'll get to those in a minute. Now, in yeast, there is only one pathway, and that is the pathway that stems from an ER stress sensor kinase called IRE1, which sits in the ER membrane, whose downstream substrate is the transcription factor XBP1, and I'll say more about that in a minute. In mammalian cells, there are actually 3 different branches of this pathway, but I'm going to focus on that one particular pathway that we spent most time studying. So how does this work? IRE1 sits in the membrane of the ER as a monomer that binds a chaperone protein called BIP. And when it senses a signal of stress, and that can be from unfolded proteins in the ER, or it can be from external stimuli like hypoxia or a low pH, it dimerizes, and its kinase domain autophosphorylates. And that autophosphorylation leads to the activation of the second important domain, which is called an endoribonuclease domain. And that RNase domain does an unusual event. It splices out 26 base pairs of what we call the pre-XBP1 mRNA. And this is the mRNA that is transcribed from the DNA. It clips out 26 base pairs of that, puts it into a different reading frame, and that makes it become a very potent transcription factor, which then goes into the nucleus and binds to a whole series of its target genes. And while these target genes can differ from organ to organ, as we showed over the last couple of decades, in common, it induces the expression of chaperone genes and foldases. It controls ER-associated degradation, and it serves as quality control. It also represses autophagy, and it controls, in part, amino acid metabolism. So that's what we call the spliced or mature form of XBP1, a potent transcriptional activator that has a whole series of genetic targets. And many of those targets are in professional secretory cells, and over the years we've been able to show that there are certain cells that cannot develop in the absence of this pathway because they cannot handle the load of unfolded proteins, and so they undergo apoptosis. The plasma cell is one of them, the beta cell, fetal hepatocytes, pancreatic acinar cells, and so on. But a graduate student in my laboratory wanted to ask whether there was a role of XBP1 in cells that weren't continuously producing heavy loads of protein, but only had transient bursts of production during differentiation. And Sarah Madigal decided to look at granulocytes because these proteins are well characterized, and they also fall into this transient burst or synthesis category. There's 3 cells that I think about: eosinophils, basophils, and neutrophils, and I put this section in here because Bill did some beautiful work on basophils and was always very interested in the type 2 response, which is driven, as you know, by interleukin-4, interleukin-5, and interleukin-13. And so I thought that he would have appreciated hearing this little story. Eosinophils and basophils, as you know, are important in asthma and allergy, parasite infections, and they develop in different stages. So Sarah showed that if you looked at eosinophil differentiation, you could see the onset of activation of this pathway in the very early stages of development. So she created animals who lacked this pathway, either the upstream sensor IRE1 or its downstream subset XPP1 in hematopoietic cells, and lo and behold, discovered that there was a complete absence of eosinophils in mice that lacked either IRE1 or XPP1 in hematopoietic cells. So no eosinophils in these animals. And I thought that was particularly interesting because this wasn't true of basophils and it wasn't true of neutrophils, cells that also have granules and are under very strong stress. So she showed that the eosinophil development was arrested at a very early stage of eosinophil differentiation. And you can see that here by looking at the early stage, the GMP, which under the influence of the transcription factor GATA2 transits to an eosinophil progenitor. And it is at that stage that you lose the expression of eosinophils. And then you move on to an immature eosinophil under the direction of another transcription factor called GATA1. And if you look by EM at eosinophils from normal animals and XPP1 or IRE1 deficient animals, you can see that they have a very dilated endoplasmic reticulum and they have very poor granule formation. So Sarah put it together this way. In normal animals, what happens when you develop eosinophils is you get the induction of GATA1, important transcription factor, and that leads to the onset of granule formation. Then these granule proteins mature, GATA1 expression remains high, and the granule proteins are packaged safely into secretory granules where they can be secreted, and there you have a nice, mature, beautiful eosinophil. But if you don't have this particular branch of the ER stress pathway, while you get GATA1 induction and the onset of granule formation, the cell can't handle the processing of those granules, and they can't increase in size and be packaged. At the same time, for reasons that we don't understand yet, you get downregulation of this critical transcription factor GATA1, which controls the expression of a lot of eosinophil terminal differentiation and effector genes like myelin basic, like eotaxin and others, and MBP, that are critical for the function of EOs, and so you basically lose all your eosinophils. So thanks to Bill's influence, we were able to identify the first factor that's specific for eosinophil development among granulocytes and show that in addition to its function in many continually protein-secreting cells like pancreatic acinar cells and fetal hepatocytes, it's also required for some cell types, but not others, that undergo these transient protein secretory bursts. Well, that was the story, and I think those three very brief stories speak to three of Bill's contributions and his influence on my career that he made by his wonderful, wonderful discoveries in those fields. And now we come to the latest adventure that we've had in the laboratory, and that is the function of this pathway in cancer. And we spent a lot of time over the last couple of decades systematically deleting this pathway in vivo in a mammalian organism because almost all of the work had been done in yeast. And we had a number of interesting discoveries, but the one that I want to tell you about today is the function of this pathway in cancer. Now, we had shown actually about a decade ago, at the same time that we discovered that XPP1 was vital for the formation of plasma cells, the postdoc in my laboratory at that time, Anhui Li, said, well, maybe it's important in the malignant counterpart of the plasma cell, the multiple myeloma cell. And so he did experiments and showed that if you silenced this pathway in multiple myeloma cells, they died. So we certainly had some hints that this pathway was going to be important in the tumor cell. And other laboratories confirmed those data and extended them. Most recently, it's been shown that XPP1 is important in CLL, in chronic lymphocytic leukemia. So I'm going to show you just one slide that summarizes the data that another postdoctoral fellow in the lab, Zi Chen, published in Nature about a year and a half ago, 2 years ago, where he looked at triple-negative breast cancer. So, we're going to talk about tumor cell intrinsic IRE1 signaling in just very briefly, and then we're going to move on to how XBP1 also functions in the immune system. But let's talk about breast cancer first. And I think, as everybody knows, triple-negative breast cancer is a highly aggressive breast cancer. It's about 20% of all breast cancers, and it occurs, unfortunately, often in young women. To date, we don't have any targeted therapeutics for breast cancer. We have chemotherapy, but once relapse has occurred, triple-negative breast cancer is very difficult to treat. And so Zi set out to look at the function of IRE1 or XBP1 in triple-negative breast cancer cell lines, and I want to stress that he also did this in primary tissues from patients with triple-negative breast cancer. It's important not to rely just on breast cancer cell lines, but to show that this also occurs in primary tissue. And he did the typical xenograft experiment where he silenced XBP1 in these cells, either in the primary cells or in the breast cancer cell lines, under an inducible promoter, transferred them into the mammary pads of immunodeficient mice, and then fed the mice after the tumors had grown to a certain level with doxycycline, which activates the CRE and deletes XBP1. And in contrast to the mice that had received a control scrambled siRNA, where tumors developed, and you can see that in the upper panel here, and metastasized to the lung, the tumors in those animals where the tumors had been silenced of XBP1 expression, the tumor persisted at the same level and eventually disappeared. Now, I think even more important than that, as I mentioned, when chemotherapy fails, there's nothing else at this point, or there are some things in the pipeline, I shouldn't say there's nothing else. There have been some exciting advances recently. If you silence this pathway at the same time that you administer 2 different chemotherapy agents that are used in triple-negative breast cancer, doxorubicin and paclitaxel, you can prevent the relapse, at least in this animal model. And perhaps most importantly, by doing transcriptomic and ChIP-seq analysis, Zi was able to identify what we call the XBP1 fingerprint, a group of genes that indicate that that cell is highly expressing this program, this particular ER stress program. And what we found looking at 2 independent cohorts of patients with triple-negative breast cancer, that the prognosis of these patients correlated with the expression of high levels of this particular ER stress pathway. We then turned our attention to XBP1 in the immune system in the setting of cancer. So we had looked at the function of XBP1 in immune cells over the last decade and showed that obviously it was critical in the B cell. It was also important in the macrophage in the setting of infectious diseases like F. tularemia, and Vanya Lazarevic, who's in the audience here, is now faculty here at the NCI, did those experiments and showed that in the absence of this pathway, there was a defective secretion of 2 important cytokines, IL-6 and TNF-alpha. And so the mice infected with tularensis, pneumonic tularensis, died. But we had never looked at it in the setting of cancer. And I have to say that I was stimulated to start looking at cancer for partly a personal reason, because around that time when we started to look at cancer, my mother had died from lung cancer. And, you know, I think this kind of personal experience can influence one's work, and it did in my case as well. And so I thought, let's look at whether anything's happening in cancer. So I'm just going to give you, the audience, a brief, very brief summary of cancer immunotherapy to make sure we're all on the same page. And the ultimate effector cell in the many approaches that we use for cancer immunotherapy is really the T lymphocyte. But activating the T lymphocyte, you can get there in many different ways, as we all know. it had been shown many, many years ago that the correlation between T cells infiltrating a tumor, this happened to be colorectal cancer, and the expression and the presence of type 1 immune T-bed expressing T lymphocytes correlated with a good prognosis. So, and that's been shown many, many times, that tumor infiltration by T cells is a critical measure of the immunogenicity of that tumor and likely its response to therapeutics. Now, cancer immunotherapy and attempts to make your own immune system wake up and kill your tumor have been going on for over 100 years, started with a surgeon about 100 years ago named Dr. William Coley, who noticed that when he operated on some of his patients to remove the cancer and those wounds became infected, that those patients' cancers often disappeared. And so he hypothesized that there must be some endogenous system that was responding to the bacteria or whatever was in that infection to activate your innate and adaptive immune systems, although the innate immune system hadn't been discovered yet, so to activate whatever it was that was killing off the pathogens to also kill off the tumors. And he actually bottled this up into a little medicine called Coley's toxins, which was distributed for quite a while until it was replaced by the advent of radiation therapy and chemotherapy. And others, like Steve Rosenberg here at the NCI, made significant contributions to looking at activation of T cells and transferring in activated T cells and interleukin-2 into patients with melanoma, and saw some success. But I think the tipping point came from Jim Allison, who won the Lasker Awards just a couple years ago for his contributions to the notion that rather than directly activate T cells by activating stimulatory receptors, one should inhibit the inhibitory receptors. And the particular receptor he looked at, of course, was CTLA-4, whose ligands are CD80 and CD86. And subsequently, another inhibitory receptor, PD-1 and PD-L1, have been the target of cancer immunotherapies, which I think broadly, and this is obviously simplistic, fall into three main buckets. So the most successful, because it's the most far advanced, is checkpoint blockade, and that's the administration of antibodies that block CTLA-4 or block PD-1 or PD-L1 in patients. And those have led to some very, very impressive responses. The second bucket, I would say, is vaccines, and as Harold pointed out, we have two vaccines against HPV and against HBV that have been shown to be very powerful in preventing cancer, if only people would get vaccinated. This field is otherwise, I would say, quite in its infancy, although I know that Jay Brzozowski is in the middle of doing a trial with a putative vaccine, and so far looking good, so we'll keep our fingers crossed, Jay. And I think it's a very active field. There are many companies that are involved in this process as well, but I think we would all love to be able not just to treat cancer, but to prevent it so that our children and grandchildren can be immune from it. And of course, adoptive T-cell therapy, which I won't talk further about, but has been pioneered by Carl June and Michelle Sadaline and others, where T-cells are activated in a dish and then transferred into patients after they have been transduced with the appropriate T-cell receptor. And this has worked best in hematopoietic or liquid tumors, has not yet worked in solid tumors, and does have significant toxicity. But we have to recognize that we have a long ways to go, because these breakthrough immunotherapies actually only work in some tumors. They work in melanoma, non-squamous and squamous lung cell cancer, head and neck cancer, bladder cancer, kidney cancer, and Hodgkin's lymphoma. But in those cancers, they only work in a minority of individuals. If you combine PD-1 and CTLA-4 blockers, then you can increase the rate of success, but it's still only 40%. And there are some tumors that are completely resistant to checkpoint blockade: prostate cancer, pancreatic cancer, ovarian cancer, glioblastoma. So we need to understand how to get to the place where we can treat 100% of patients who have every different kind of cancer and with immunotherapy. And our postulate, and many others, we're not alone in this, is that the current immunotherapies fail in part because these tumors are surrounded by a highly immunosuppressive barrier. If you take a biopsy of a pancreatic tumor or an ovarian cancer, you'll see that the majority of the cells in some cases are not the tumor cells. They are a cluster of immune cells that are highly immunosuppressive. And if this barrier exists, then you can activate T cells all you want, but they can't get in. So there are myeloid-derived suppressor cells, prostaglandins. There are these various inhibitory checkpoints. There are T regulatory cells. And there are a lot of immunosuppressive cytokines that are secreted by the tumor itself and by the surrounding cells. And there's also significant dendritic cell function, which is what I'm going to talk about. So when you think of where the current immunotherapies act, vaccines obviously act at an early stage at the dendritic cell and antigen presentation. Adoptive T cell therapy is clearly putting in those active T cells right away. And of course, checkpoint blockade is a way to activate the endogenous T cell. So we would say that there's really a fourth bucket, which has been very much underexplored, and that is this very immunosuppressive tumor microenvironment. Now, there are some strategies for targeting the TME, but they're quite narrowly focused. Interesting, I think most interesting may be inhibitors of arginase or IDO1 inhibitors, and you can see some of these are preclinical and some of them are in early phase trials. So there are some therapeutics out there, but we suggest that from the data that I'm going to show you, that targeting this particular branch of the ER stress response might be able to reprogram the entire tumor microenvironment. And why do I say that? Well, first of all, let's think about the stimuli that are going to activate ER stress in the setting of cancer. You've got a very hostile environment. You've got a low pH, hypoxia, so the tumor cells are not getting enough nutrition. They're trying to take up a lot of glucose. And you have flooded with reactive oxygen species. Chemotherapy and radiotherapy also evoke ER stress. And then, of course, there's a whole other issue of high metabolic demand, both in the T cell and in the tumor. Now, we think the UPR via this middle branch, IRE1, can help with this by controlling protein folding or by other means. So the question that Juan Cubillos Ruiz asked when he came to the laboratory several years ago as a postdoctoral fellow was, what about XBP1 and IRE1 in the immune system? Now, we weren't terribly surprised that targeting the tumor itself in triple-negative breast cancer was effective because we had shown earlier that it was effective in myeloma. But I must say, my prediction was that these experiments would show that crippling IRE1, XBP1 in immune system cells, dendritic cells or other cells, would actually blunt the immune response. But I was, as usual, completely wrong. So Juan Cubillos Ruiz took this on, and I'm going to talk about briefly his work in dendritic cells. Juan got his PhD with Jose Conejo Garcia at Dartmouth, where he studied ovarian cancer, which, as we know, is a very lethal cancer in women. 1 in 95 women will die of ovarian cancer. Now, if you can pick ovarian cancer up at a very early stage, before it has broken through the ovary and metastasized, you have a very good chance of survival. But as we know, that's the minority of patients. Most of the times, tumors are detected at the late stage where metastases have occurred to the peritoneum and to other organs, and there the survival is really very dismal. The length of survival has increased, but the ultimate outcome for most women with ovarian cancer is not good. And if you look at the 5-year survival rate, you can see it really hasn't changed in the last several decades. Now, if you look at the cells that are present in either human or mouse ovarian cancer, you can see that there are tons of CD4 positive, CD45 positive leukocytes, and there's a huge number of these very immunosuppressive dendritic cells. if you can take them out in a dish and try to activate them and then put them back together with a T cell, they can activate a T cell, but in the native environment of the peritoneal cavity, they are highly immunosuppressive. So the first thing Juan did was say, well, is this true in humans as well? And luckily we had wonderful collaborators. The head of gynecologic oncology at Weill Cornell, Kevin Holcomb, provided us with many, many samples of ovarian cancer ascites and ovarian cancer tumor cells as well from the many patients he saw. And what Juan saw immediately was that there were very high levels of this spliced XBP1 protein in these dendritic cells from the ascites or from the tumors themselves in virtually every patient with ovarian cancer. And consistent with that, the universe of target genes that are activated by XBP1 were elevated in those dendritic cells as well to show that this was functional XBP1 protein. It was turning on a genetic program. So Juan looked at a mouse model of primary ovarian cancer, which I won't detail except to say that it bears the two most common mutations that are seen in human ovarian cancer, KRAS and P53. And this is a local model in which you can actually selectively delete KRAS and P53 in one ovary but not the other. And when you do that in an animal whose dendritic cells lack IRE1 or XBP1, the tumor does not grow very well. And you can see that here. He also looked at a metastatic model of ovarian cancer, and this is via the injection of a cell line derived from a patient with ovarian cancer that's been genetically manipulated to be very aggressive. And he compared the response of normal animals and animals that had deleted XBP1 selectively in dendritic cells and was able to show that these animals were protected from developing peritoneal metastases. They had less accumulation of ascites, they had less splenomegaly, and most importantly, they had increased survival. And they had increased survival. Again, here's the endpoint. The endpoint is the activated interferon gamma secreting TBAT positive type 1 T cell. And if you looked at any of these models, and he did another couple models as well, you could see the production of type 1 T cells secreting lots of interferon gamma and granzyme B and other cytotoxic molecules. So you can delete it in the dendritic cell, and ultimately that leads to the ability of effector T cells infiltrating the tumor and killing it. So we asked ourselves, how does this happen? Why does deleting this particular pathway in a dendritic cell activate the immune system through T cells? And so we did the usual transcriptomic analysis of the dendritic cells in this setting, and we found 3 pathways that were very activated in immunosuppressive tumor-associated dendritic cells. Not surprisingly, one of them was, of course, the ER stress pathway. A second was a pathway in carbohydrate metabolism, and that was consistent with a wonderful paper from Phil Shearer's lab a couple of years ago showing a role and a function for this pathway in carbohydrate metabolism. And finally, lipid metabolism. Now, this should not have been a surprise to us because we had accidentally discovered a function for IRE1 and XBP1 in hepatic lipogenesis, which is a big surprise to us, as I must say many of the discoveries we've made have been a surprise to me. Hui had set out to ask what the function of XBP was in mature liver, since it was so important in fetal liver. And he came back very discouraged because it looked like it had no function in adult liver. Levels of proteins were normal, the animals looked normal. And I said, well, why don't you go to the clinical chemistry laboratory and check everything you can? And he came running back, and he said they have very low serum cholesterol and triglycerides. So it turned out that XBP1, apart from its function in the ER stress response, also controlled the expression of genes encoding enzymes that are involved in hepatic lipogenesis, like SGT1 and DGAT and others. So it controlled lipid metabolism. And then we remembered a couple of very good papers from Dmitry Gabrilovich's laboratory where he showed that accumulation of lipids in dendritic cells led to their dysfunction. And that in particular, oxidized lipids could block antigen presentation. And so we looked by EM at our dendritic cells, and sure enough, the lipid droplets that you can see in the red arrows that are pointing to them in the normal animal, normal dendritic cell, full of lipid droplets, but if you delete this pathway, no lipid droplets accumulate, and that's quantified in the panel to your left. So how does this work? Well, the hypothesis that we came up with and were able to validate is that there are many factors, there are many substances that are produced in the tumor microenvironment, such as reactive byproducts like 4-hydroxy nonanol acid, and that these substances are taken up by the dendritic cell. They are lipid peroxidated by reactive oxygen species, and they generate these highly reactive aldehydes. And these aldehydes then bind to proteins in the ER of the dendritic cell. And schematically, it looks like this. There are known and unknown tumors that are floating around in the ascites, in the tumor microenvironment. And they're taken up, they undergo lipid peroxidation, highly reactive aldehydes, and they actually bind, and we show this by mass spec, to these important chaperone proteins that are in the endoplasmic reticulum. And that misfolding and of those proteins, the alteration in the structure of those proteins then activates the unfolded protein or ER stress response. And so you activate IRE1, which activates XBP1, and XBP1 drives lipid synthesis in dendritic cells just as it does in liver. Now, how lipids, droplets, interfere with antigen presentation and processing is still unknown, but I can tell you that if you look at the surface of these dendritic cells, they have very low levels of MHC tetramers on their surface. So somehow levels of MHC are normal, but you cannot process these native antigens and display them on the surface. And how that happens, I think we still don't understand. But indeed, if you look at these dendritic cells, you can see that they have enhanced antigen presentation capability. I showed you that they can activate T cells before. And so we asked whether we could devise therapies that might utilize this knowledge to target ovarian cancer and to activate the immune system, because none of these other approaches has worked in ovarian cancer. And what we tried first was to transfer in these XBP1 or IRE1 deficient dendritic cells to a mouse that was injected with tumor. So could they act as a dominant force to lead to the activation of T-cell immunity? And the answer to that question was yes. If you transfer in these immunoactive dendritic cells, they can overpower the immunosuppressive dendritic cells, activate T cells, and you fail to get much tumor growth. And this raises the possibility, although frankly I think it would be difficult to do, that you could genetically edit dendritic cells from patients. From ascites, you could take out the ascites, take the dendritic cells, silence IRE1, and then put them back in, just as is being done for T cells. Another approach is to deliver an siRNA for IRE1 or XBP1 directly to dendritic cells through injection, perhaps at the time of surgery after the patient's tumors have been debulked, as essentially as a neoadjuvant. And so we looked at the ability of nanoparticles that encapsulate siRNAs to affect the course of ovarian cancer progression. These are polyethylenamine particles that, for whatever reason, and it's not clear, selectively target dendritic cells. And when you do that, you reduce the expression of XBP1 or IRE1 by about 50 to 60%, but that seems to be enough to reduce the amount of ascites and to prolong survival in these animals. And this is, again, dependent on T cells because it doesn't happen if you delete T cells. Now, just a word about the function of this pathway in other cells. So we've talked about it in dendritic cells. Myeloid-derived suppressor cells are very important in creating this very immunosuppressive tumor microenvironment. And some recent work from Dmitry Gavrilovich again has suggested that one can convert a highly immunosuppressive MDSC into an active neutrophil-like cell that is anti-tumoral simply by blocking ER stress, in particular IRE1. And he was able to demonstrate this in human MDSCs by using a IRE1 inhibitor called BIO9, and showed that if you treat MDSCs with this IRE1 inhibitor, they become active neutrophils that can kill tumor cells. We also have data that suggest that this pathway is very important in MDSCs, in part through regulating prostaglandin production and also in other ways. Most recently, we have worked on the function of XBP1 and IRE1 in T cells, and it has a prominent function in T cells, but those data aren't quite ready for prime time, so I won't present them to you now. So let me summarize. We have shown that, somewhat to our surprise, this pathway is very, very important in cancer, and it's important not only in targeting the tumor itself, but also in simultaneously activating the immune system, something that we did not expect. So it can directly inhibit tumor growth. We've shown that in myeloma, in breast cancer. Others have shown it in CLL. But at the same time, you get activation of the immune response. And if you actually delete this pathway in all hematopoietic cells, you see an even more dramatic result than I showed you from deleting it just in dendritic cells. So while we believe dendritic cells are very, very important, we think that targeting this pathway will expand the ability of the immune system to kill the tumor cell. So we treat cancer today with the usual tactics: surgery, chemotherapy, radiation therapy, and more recently, with the explosion in genomics, precision genomics, there are a panel of drugs that are available that can target certain genetic mutations in tumors, the most successful of which is really Gleevec, because it can treat CML and GIST for a long, long time. Some of the other targeted agents, like Xalkori or others, suffer from the fact that the tumor is really smarter than we are, and the tumor under pressure evolves and generates other genetic mutations. And so I think these patients obviously develop resistance, and I think the next frontier in precision genomic medicine and targeting the tumor itself is going to be figuring out how to circumvent the resistance. And I think that's probably going to be both by looking at the effect of the tumor on the immune system and the effect of the immune system on the tumor. And today we can also activate the immune system, certainly through checkpoint blockade, but as I said before, we have a long ways to go to be able to treat all tumors that way. So I hope that 5 years from now, if we can accelerate our therapeutics from 10 years down to 5 years, as the Vice President has suggested in the Cancer Moonshot, that we will be able to make some cancers at least chronic diseases, and perhaps even to cure some cancers. But I think it's going to be like HIV/AIDS, where the breakthrough came in part because the NIH funded some really good basic science research in virology, studying the virus and figuring out how to attack it, and making the discovery that if you attacked it with several drugs at the same time, you could prevent the virus from mutating as quickly. Now, cancer is much more complicated. There are hundreds of different kinds of cancers and with hundreds of different kinds of genetic mutations. But I think, again, it's going to be a matter of figuring out which therapies one can combine without too much toxicity. And it's going to be a combination, I think, of immunotherapies plus other immunotherapies, targeted agents plus other targeted agents, and then the whole mix together. That's going to take a long time. That's a huge matrix. And so we're going to need everybody's help in figuring out how each tumor, because each tumor is different, how we can treat those tumors most effectively. And to me, the biggest gap we have is figuring out how to do that kind of combination therapies ex vivo in human cells so that we don't have to do clinical trials, the thousands of clinical trials, testing out all these various combinations. We have to figure out, either using organoids or other systems yet to be discovered, how we can most quickly evaluate each patient's tumor response to a variety of different immunotherapeutics and targeted therapeutics. And I want to end there, and thank you very much for listening to me. I particularly want to call out Juan Cubillos-Ruiz, who's now an assistant professor at Weill Cornell. He's responsible for the work on cancer immunotherapy. Zi Chen, who did the experiments on targeting the tumor itself. Sarah Bedegal for her work on the function of IRE1 and eosinophils. And Anhui Li, who has over the years contributed enormously to this field. And I thank you very much, and I wish Bill were still here with us. Thank you.
I have simple question, but I need to explain the reasoning. Did deficiency SPP1 also resulted in deficiency of adenosine-generating CD73? And the reason I'm asking you, because about a year and a half ago, you published, I believe, Nature, that SPP1 controls HIF1 alpha. And HIF1 alpha upregulates all known adenosine-generating enzymes, CD73. And triple-negative cancer, the worst ones, which also resistant to anthracyclines, are resistant that only because they upregulate adenosine-generating CD73.
Yeah, that's a great question.
And then synthetic antagonists of A2 adenosine receptor make these untreatable tumors treatable. That means implication is that XPP1 was something that was looking forward to something like that, because HIF1 alpha, hypoxia-inducible factor, wrong name. It's a hypoxia-stabilized factor. But this XPP1 is expressed in many and not only hypoxic areas. That will explain all all resistant tumors that now could be treated.
Well, we should try that combination of IRE1
Thank you very much.
and adenosine. You're right. XPP1 interacts, binds to HIF1 alpha, and serves as a co-activator. And that's really how it drives triple-negative breast cancer. And it affects the expression of HIF1 alpha target genes. I can't remember if CD73 is one of them. Sure. I wouldn't be surprised if it were, but sounds like a great new experiment to do.
Let me just make a very quick announcement, which is that there are CME credits available for this if you sign outside, and also the reception afterwards will be in the same place as the earlier coffee breaks. Rachel, go ahead, and then Jay.
Laurie, wonderful talk. So my question is, as of course we all know, cancer and autoimmune disease are two sides of the same coin. So I am wondering if it is possible, if your lab is exploring targeting these pathways in immune cells in order to address issues in autoimmune disease.
So we have done a little bit of that, Rachel. We deleted the pathway selectively in B cells in a mouse model of lupus, a disease that, as you know, is driven by bodies. This is Betty Diamond's model of anti-DNA antibodies, and these animals usually get cerebritis and not surprisingly, when you don't have bodies, as you don't when you delete XP1, you don't get the disease. So yes, I think there's some potential value in that.
Thank you.
Laurie, very elegant, beautiful story. In some of Dmitry Gabrilovich's earlier work, he had shown that STAT3 played a critical role in diverting dendritic cell precursors into MDSCs and preventing maturation into mature DCs. So how does the XBP1 pathway and the lipid pathway that you described interface with STAT3? Are they two independent mechanisms, or do the two interact in some way?
It's been suggested by Dmitry and by another group that STAT3 might be downstream of IRE1. And this is something that we haven't looked at ourselves, but are aware of that, and it would be very interesting if that were the case.
Okay, very nice.
Beautiful talk, Laurie. Bill would be proud. As you know, there are many small molecule inhibitors of the unfolded protein response, which work at different points in the unfolded protein response. I know, for example, that metformin has been shown to have anti-tumor activity in some animal models. And that one works probably by activating AMP kinase. Have there been any experiments done to look to see whether relatively smaller concentrations of several UPR inhibitors can be used together in the hope that maybe you'd achieve the same goal with less toxicity?
So there are a number of IRE1 inhibitors. Unfortunately, none of them is very active in vivo, and several of them are actually non-selective. I'm thinking of one in particular that looked to be very potent, but when we tested it, turned out to be IRE1 independent, so that it mediated the same kind of suppression in cells that lacked IRE1. So we have to look both for potency and selectivity. In the interest of transparency, we have formed a small company called Quintus, which is dedicated to looking for small molecules that will inhibit the kinase domain of IRE1. And who knows, there are a number of companies working on this, and I very much hope that one of us at least will be able to find a small molecule that is potent, that is active in vivo, and that has fairly selective effects. But you raised the point of, would we use an IRE1 inhibitor for a million different diseases? Would we actually use it to lower somebody's serum cholesterol and triglyceride? And I think from all that we've learned from deleting XBP1 or IRE1 selectively in different organs, whether it's the brain or the pancreas or the liver or the cartilage, whatever, that it's probably not something you would want to use chronically. For one thing, you'd have very few antibodies if you did that. So I think it's a pathway you'd want to use in an acute situation like cancer, like metastatic disease, where you could give intermittent doses of it.
Excellent talk, and I've always been a big fan of your work. I've been curious as to whether anything is being done right now to prevent the cancer from coming back after it's already been dealt with in 5 years when we reach that point. Is there any current study being done to prevent it from coming back over and over and over again after it's been eradicated?
That is a big question, and I think that much of the work that's going on in cancer that applies to treating active cancer would also apply to the prevention of recurrence. If you believe in the cancer-initiating stem cell, that's a population you need to wipe out, right? And there are a lot of laboratories working on how you get that quiescent stem cell and actually eliminate it, because it's pretty hard to do. We all know individuals who have had breast cancer, have been cancer-free for 20 years, and the cancer comes rip-roaring back after 20 years of being silent. It's not so different from the latency that you see with HIV in that sense.
One thing I was curious about is that if modification of the immune system, like we're doing for curing of cancer, could potentially posit a potential avenue for preventing it from coming back.
One could hope.
Yeah.
Thank you.
Let me thank Lori once again, and really all the speakers from the entire day. I think that Bill, as people have said, would have been thrilled with this day in his honor, and to have heard all the great research that's been presented by people who work with him. So thank you very much. There's the reception, as I mentioned before.